All right, let's go. So what we're going to focus on today, right? You can see the title. And again, this is, the chapter from your old textbook, the textbook I used to use. But application and utilization of immune responses, right? So we're going to focus really heavily on the concept of immunity. And when we talk about immunity, right, you'll see there's different versions, but one of the things that we'll spend a lot of time on is this idea of vaccines. And towards the end of the chapter, we'll touch on some other research laboratory-based tests that utilize concepts or parts of the immune system. But so this is definitely kind of vaccine heavy. And so when we talk about the principles of immunization, we're going to talk about different types of vaccines. We'll also touch on the importance of immunization. We're going to talk about some of the different diseases and illnesses that we're still trying to develop vaccines for, some of them that we've successfully eradicated because of vaccines, and then immunological testing. This is kind of the research-based approach to using immunologic principles to help us learn about things that we are trying to study or understand. So what I'm going to do here is just show you guys a couple of videos. Some of these videos are serious. Some of these videos are funny and serious, I guess. So, just enjoy some of these. Let's see, so I think this is the one
like we're headed in the wrong direction. I want to get serious for a minute if I could, because I've been hearing a lot of talk lately, and I don't know if this is more prevalent in LA than other places, but I feel like it probably is. But there's a small but still sizable group of people who are choosing not to vaccinate their children. Here in LA, there are schools in which 20% of the students aren't vaccinated because parents here are more scared of gluten than they are of smallpox. And as a result, we now have measles again. We've got measles. I want to say something about it. I know if you're one of these anti-vaccine people, you probably aren't going to take medical advice from talk show hosts, and I don't expect you to. I wouldn't either. But I would expect you to take medical advice from almost every doctor in the world. See, the thing about doctors is they didn't learn about the human body from their friend's Facebook page. They went to medical school where they studied all sorts of amazing things, like how to magically prevent children from contracting horrible diseases by giving them a little shot. You know those little shots of Botox? Which is botulism, by the way. You get in your face to make your head look smooth and your eyes look crazy. A little shot like that and poof, polio is gone. But some people do not buy into that because they did a Google search and Jenny McCarthy popped up and she had clothes on, so they listened to what she had to say and decided not to vaccinate their kids. Now, and by the way, I want to say, this would all be okay if your kids were the only ones affected. They're your kids, but they're not, because unvaccinated kids put all children in danger, especially babies who are too young to get the vaccination shot. But of course, that's according to doctors, so, take that with a grain of salt. Now, some people are saying kids who aren't vaccinated shouldn't be allowed to go to school or daycare or go to public places, but here's where I think it should go. If you are one of those people who knows more than doctors, that's fine, no vaccinations, but you're not allowed to go to the doctor anymore. Okay? Why would you even want to go to a doctor who knows less than you do? So, if you fall and you cut your head open, you will not be admitted to the emergency room. Stay at home, find that sewing kit you stole from the Marriott, and stitch it up yourself, Doc. The doctor will see you never. Oh, by the way, you should also let your kids smoke. Why wouldn't you? The only people who say not to are doctors, and they don't know. So I feel like this is starting to snowball. So we invited some real doctors to address this. Again, these are not actors. These are actual medical professionals.
Okay. All right. So, I mean, I think Jimmy Kimmel's hilarious, and so I like that. And it is a funny approach. But what are some of the good points that he maybe brought up, or some of the kind of the things that we talk about when we're discussing vaccines versus the anti-vaccination movement? What are some of the? I mean, I know he was joking around, but he did bring up some good points. And so what are they? Did anyone catch some of them?
Um, I know that he said that, like, even though, like, you're not gonna get your kid vaccinated, your kid's not gonna be the only one that's affected, and your kid, like, being around other kids or even babies that can't, be vaccinated is, like, you're affecting them too, so it's not just, like, your child.
Perfect, right? And that's really good, and we'll touch on this again. But what he's referring to there, right, is this concept of herd immunity. And the idea in herd immunity is there are some people that can't get vaccinated, right, for really legitimate reasons. For some vaccines, right, children are just too young. You have to hit a certain age before you can get that vaccine. Um, Older folks, right, sometimes cannot be vaccinated, or the vaccines just don't work in them as well. Pregnant women, people that are undergoing chemotherapy or radiation for cancer, or people that have a certain autoimmune disease sometimes can't be vaccinated. And so, what Jimmy Kimmel was saying there is that, everybody that can get vaccinated, if they do get vaccinated, and again, for different diseases, there's kind of different thresholds. But if enough of the population is vaccinated, the disease can't spread in that population, and then it's going to protect those people that aren't able to be vaccinated. Right? Did anyone else notice anything else interesting about what he was saying, although in a joking manner? Nothing. Does anyone use social media? No, you don't. Caroline, you don't use social media at all?
Oh, I do.
Yeah. Do you think that social media is the best place to get information?
Definitely not.
And so I would totally agree. And I want to kind of touch on that because I actually don't think social media is bad. Where social media gets dangerous is if Caroline reads a post and it says, all crows carry rabies, which would be interesting, right? Because rabies is spread by mammals. But so she reads that, and then she goes outside and she starts shooting all crows, or she develops an abnormal fear to crows. And I'm not even saying that there's definitely not truth to that post, right? But a lot of times when we're using social media as an information source, we don't usually follow up, right? That information. And so when it comes to vaccinations, um, there's a lot of times that there's not follow up based on certain posts, right? And so I think where we get our information and how we consume our information is really, really important. All right. And so that's what Jimmy Kimmel was trying to report. Does anyone know? He mentioned Jenny McCarthy. Probably most relevant right now if any of you watch The Masked Singer on Fox, right? She's one of the panelists on there. Does anyone know um, why she's mentioned as part of the anti-vaccination movement? Anyone know? Any of my pop culture gurus? Does anyone know who Andrew Wakefield is? Has anyone heard of you do, Lauren? Who is he?
He wrote the Lancet paper that pretty much started the anti-vax movement.
All right, so yes, the kind of the modern anti-vaccination movement as we know it, right? Andrew Wakefield published a paper in Lancet trying to link, I believe it was MMR, measles, mumps, rubella vaccine, to developing autism, right? And so Jenny McCarthy's son has autism. And so a while ago, and this is probably 10 plus years ago now, 10 to 15 years ago, interesting, when she was dating Jim Carrey, that was a fun combination. But they were huge in the anti-vaccination movement and were very public and very vocal, right? So that's the reference that he was making in that video. Okay, cool. So let's keep going. We will look here at some more serious news reports in just a second, but I think it's fun to share truth. I think with a little bit of humor, and you'll actually see in one of these upcoming clips. He was kind of making fun of Beverly Hills and Hollywood, and they actually are one of the hotter spots of the anti-vaccination movement. Okay. So when it comes to immunity, right, we have again kind of two major branches, right? I love my two major branches because then I can just form two major branches off of them. But we have, all right, when we're talking about immunization, we have natural acquired immunity and we have artificial acquired immunity. And without even reading these definitions, you can probably guess, right, which is which. And so if you've ever sneezed on somebody, been sneezed on, coughed on somebody, been coughed on, right, fun stuff like that, right, that is a natural acquisition of an infection, which could hopefully lead to, right, a response by your immune system that may or may not develop immunity kind of down the road, right? So you are naturally encountering that pathogen, you develop a resistance to that pathogen, and then that resistance lasts for however long, right? And again, resistance to pathogens can vary depending on mode of delivery and type of pathogen, right? But the immune system is educated through natural events, right? You come to contact with somebody, you come to contact with it in the environment, and so you develop immunity that way. Artificial acquired immunity, right, is basically our attempt to mimic a natural event. And in some ways, I like to think of it as almost tricking the body into responding to a pathogen that is not actually pathogenic or is not nearly as pathogenic as it would be naturally, right? So you're mimicking natural events to educate the adaptive immune system. So this is what we do when we vaccinate, all right? You are making the body think that it's being infected, right? Or that it's seeing this infection naturally. It responds, and then you're immune to that pathogen. All right. So again, we can further break down natural and artificial into active immunity and to passive immunity. So natural immunity, all right, or natural active immunity is an infection, right, that you get from interacting with a person or interacting with an environment, all right? So it's usually a robust response because your whole immune system is involved. If, right, you work your way through the infection, there's a chance that you may have immunity. Now, again, one of the things I want you to think about, and I'm sure you're aware of this because you're like, well, why do I get the cold every year? Why do I get the flu every year? Why do I get the flu vaccine every year? Right? Some pathogens are really, really good at changing, right, and mutating and adapting. And so it requires, right, a new generation of a response every time you see that new version. Artificial active immunity, right? This is a vaccine, all right? So again, it's artificial, but right, it induces a hopefully robust immune response and it induces immunologic memory, right? So that's one of the things that I really want to highlight here too is active immunity equals memory, right? You remember we talked about clonal selection and expansion. You have those effector cells that fight the disease, but then also, right, during that division process of those cells, you have some that become memory cells and are waiting around to help you fight that infection later. So even after you clear that infection, these cells remain so that you could fight it in the future, right? And this differs from passive immunity. So passive immunity, right, again, has a natural and an artificial route. In this case, right, you typically are passing along antibodies, right? So, here it shows a mom and a nursing baby, right? You could also say this is the case for a mom and a baby that's in utero, right? That mom is sharing antibodies with that baby that help shape its immune system, help protect it until the baby's immune system is fully functional, right? So this is natural passive, right? This is going to be between mom and baby, right? Artificial passive immunity, right, is basically when you take antibodies that have been generated by someone else or something else, right, and inject them into an individual to protect them, right? So those of you, has anyone ever been bit by a venomous snake? No, not yet. Okay. Well, if you ever are bit by a venomous snake, and I guess hopefully that doesn't happen, right? You will get anti-venom, right? And that anti-venom is actually antibodies, right, that have been generated. I think they're typically generated in a horse, um, and then they're isolated, and then they can be injected into you, all right, to help counteract the actions of that venom. If any of you have, I think I mentioned this maybe last class, stepped on a rusty nail or gotten a bad cut and they think you may have tetanus, right? You might get a little booster that is full of antibodies to the tetanus toxin. And then I don't know if you've heard of this yet, but it's convalescent plasma. Has anyone heard of that? Does anyone know what I'm talking about when I say that? Has anybody heard of what they're trying to do with antibodies to treat COVID patients?
Are they trying to collect blood from people who have antibodies to COVID-19 and then transfer it to people who are infected?
Perfect, right? So convalescent plasma is this idea of, you can take whole blood and then isolate the plasma, or you can just take plasma. But yes, so you take someone that survived COVID-19 or the COVID-19 virus, and then you give, right, that plasma that contains those antibodies to someone, right, who's fighting the disease. And the idea would be the antibodies in the patient that survived will hopefully neutralize, in this case, the virus in someone that's fighting the infection. All right. And so this has been used a couple times before. So we've used it in Ebola for Ebola patients a few years ago. We actually used it for SARS patients in the early 2000s. And we've used it a couple times for some really bad outbreaks of the flu. All right. So can anyone just guess, and I know this may be a little bit of a stretch, but what are some of the issues with potentially doing this treatment method, and maybe what are some of the challenges in learning if it's effective? Let me start with this. What do all good experiments have?
Control.
They have a control. Perfect. Thank you, Laura. All right. So one of the big challenges with convalescent plasma treatment is that there typically are not good controls, right? Because that would mean that you would say, okay, I'm just looking at my screen and I see Lauren, Megan, Noel, and Laura, and I'd say, okay, we're gonna do a blind study here, and I'm gonna give Lauren and Megan. They're gonna get the plasma from a patient that survived COVID-19, and then Noel and Laura, you're gonna get control plasmid from a person that never saw the virus. Ooh, luck of the draw, right? And so, people have proposed these studies, but there's also interesting ethical kind of implications, right? And so all of our data on, this plasma therapy um, doesn't really have proper controls set up with it. And so we have seen improvements in patients that have gotten this treatment, but we're still really not sure if we can attribute, right, those benefits to the plasma treatment, right? It's not definitive, yes, yet. What's another issue, do you think?
For COVID-19, do they know like whether or not antibodies actually confer like long-term immunity?
Um, so they don't know that yet. Um, but to Megan's point, right, and this is all passive immunity in general, right? Once the antibodies are gone, that benefit of those antibodies that you injected could also be gone. All right. And so that is a concern, even for future infections for these patients. Yes, Megan's totally right. We don't know how, if people are even really immune to COVID-19. And you've started to hear, maybe if you're paying attention to the news, and I haven't seen if the percentage has gone up, but at one point in South Korea, which again I think I mentioned has been really on top of the COVID-19 virus, they were starting to see upwards of a 2% reoccurrence of infection. And so even if you have natural immunity, we don't know how long you're protected, if you're protected at all, right? You also have the issue, though, if I had COVID-19, and I'll pick on Megan again, and I survived, right? I'm good, and she's sick, and I give her my antibodies. One thing that you are doing potentially is your antibodies help, or my antibodies help Megan, so she actually doesn't generate maybe as a robust of immune response. And so after she gets well, right, my antibodies helped her, but maybe her body didn't fight the infection on its own enough that she's open for a reoccurrent infection. All right. And so there's a lot of unknowns, right? The basic principle of the treatment is sound. All right. They are able to show, right? They're able to take these antibodies from plasma, put them on a virus that's in a petri dish, right, or a test tube in a lab, and show that it neutralizes the virus, right? So the antibodies work. They're capable of interacting with the virus, which is what we want. But there's still a lot of other unknowns. One of the other issues, too, is that if I took plasma from Caroline, Professor Perry, Rachel D., Glory, and Amanda, right, the concentration of COVID-19 antibodies in their plasma could vary greatly, all right? And so, maybe Caroline and Professor Perry survived, but am I getting the right number of antibodies from their plasma to help provide protection? We just don't know, right? And so kind of our alternative to this is that once you take plasma from a patient, right, although this is kind of a big time investment, you can pull those antibodies out and you can concentrate them. And then the idea would be you go and you get a single injection of a high concentration of COVID-19 antibodies, and that hopefully confers enough protection on you, right? So it's a really cool possibility and a really cool treatment. We just don't know much about it, all right? And from a medical standpoint, typically when we think of this treatment, we don't think of it for viruses that are part of our everyday lives, all right? And so that's one of the reasons, too, that proper studies usually aren't being done, because this is a response to an emergency situation, okay? So there's interesting potential there using passive immunity to protect from COVID-19, but will that come to fruition or actually end up being a commonly used thing or really beneficial, right? It's still too early to tell, but it's a cool kind of concept. All right. So again, the whole point or purpose of vaccines, all right, is to induce active immunity. And active immunity, all right, results in memory. And that memory protects individuals along with protecting an entire or potentially protecting an entire population, right? And that's that idea of herd immunity, all right? So this idea that a critical level of the population is immune, all right, whether that be natural, right, or artificial, right? And in doing so and reaching herd immunity, susceptible individuals that we kind of have listed, right, are then protected. And for different pathogens, right, this critical level of the population, right, that needs to be reached is different. All right. So I know for like measles, if you guys watched that TED Talk video, right, that's somewhere in the 90 to 95% range. I can't remember the number that I heard thrown out for COVID-19. I don't think it's quite that high, but it's still a significant portion. Okay. Can anyone take a quick stab or guess at how the concept of herd immunity has actually helped, in some cases, fuel the anti-vaccination movement? How could the concept of herd immunity encourage some people to not get vaccines?
Haven't people said that, like, well, since most of the population's vaccinated, we don't have to vaccinate our kids?
Perfect, right? And so they clearly have an understanding, right? Or some of these anti-vaccination people clearly have an understanding of herd immunity. And so it's the idea of, well, you know, everybody's getting their vaccines, and so it doesn't hurt, right, if I don't get vaccinated, or if I don't get my kids vaccinated, right? The problem is when you know, a sizable portion of the society starts to feel that way, then you start to lose or go away from that critical threshold, right? And so in some cases, herd immunity has become its kind of own worst enemy. So let me stop this share, and then we'll go to another one. I think we'll just do one more video. You can click on these other links.
Every one of them is a real doctor, so hear them out and then decide for yourself.
I want to play Rand Paul's comments first so you can hear them and folks at home can as well.
Sure. I've heard of many tragic cases of walking, talking, normal children who wound up with profound mental disorders after vaccines.
Now, he is, you know, obviously sounding off on this, Hillary Clinton as well. Is there a danger, Dr. Gupta, I'm going to call you in this segment, of this being politicized, a medical debate being politicized like this?
Yeah, no, there's no question. I think that it's dangerous, and to the extent that there's some pandering happening, this is the sort of pandering that can be dangerous because, you know, Rand Paul's a doctor. I'm sure he looks at studies. He knows a scientific method. He says he knows of many children who have wound up with mental disorders after vaccines. You know, I don't know where he's getting that data from. And one of the old adages, Mick, in science is that correlation does not equal causation. There may be kids who have had mental disorders that have also received vaccines around that time in life. But just because those two things happen around the same time do not make one causing the other. So, again, I don't know why he's saying this. The numbers don't bear that out. The science doesn't bear that out. I'd be curious what drove those comments.
But Sanjay, I mean, I think that what he was referring to is vaccine injuries. And there are a handful of people who get vaccine injuries. It's just that happens. There, you know, I think that sometimes people doctors, pediatricians like to pretend that vaccines are 100% safe. They're not actually 100% safe. They are safe for the community. They are the right thing to do for the community, certainly. But there is vaccine injuries. I think that's what he was referring to.
Yeah, sure. He may be. I don't know what specifically was inside his head at that moment, but let me address that, because you bring up a good point. So when it comes to shouting from the rooftops that something is potentially dangerous, look at things like vaccines and then start to compare them to things that we don't shout from the rooftops on. I got a little graphic here I want to just show you in terms of injuries. You're right. Vaccines, like most things that you put in your body, we have another graphic that points this out. Vaccines, like we put most things in your body, do have some potential injury rates. For vaccines, it's about less than 1 out of 1 million doses are you going to have a serious allergic reaction, could cause a seizure or something like that in a child. Take aspirin. This is something that we can all relate to. It's in all of our medicine cabinets. 12 events per 10,000 people could cause a potential intracerebral hemorrhage. I'm not trying to frighten you away from aspirin. Point is, the numbers are still very small. With vaccines, it's exponentially smaller risk than aspirin, and yet we're not screaming about aspirin. So that's part of the issue here. So you're right. There can be problems, but not to the degree
Sanjay,
you think.
I think that let's flip it. I think that you should be screaming from the rooftops and more. I don't think that we should be engaging people in this paranoia. The science isn't there. The practicality isn't there. I can't have my kid bring peanuts to school, but a parent has the choice whether or not to vaccinate their kids and then send them to school. Are we giving too much deference to the other side here when there really isn't one?
I agree with you, Chris. I don't think there is an other side here, frankly. We get so used to this discussion about this side and that side. The two sides could just be simply the right side and the wrong side. We don't know what causes autism. I mean, that's fair to say. We're not sure as a scientific community what causes autism, but we know that vaccines do not.
Right.
And I think it's just important to say that as succinctly as I just did, and not to say, look, I'm going to delay the vaccines. You hear doctors coming out and saying, it's okay, let the parents choose. Of course, I'm a parent. I have 3 kids. I made that choice. I got them vaccinated on schedule, on time, to protect their health and to protect the health of the people around them. I also want to show you one other thing here, and this is a little bit of a nuanced point, this other graphic I have, looking at the way that we used to vaccinate in the past, how we vaccinate now, and look at how autism rates have sort of changed since that time. If we can put that graphic up. But basically, it's showing that in the past, look back there in the 1960s, we gave a lot more in terms of vaccine load to kids' bodies. Look at 3,500 times more. And then at some point, sort of in the late 90s, we started to give lower doses of vaccines to try and get the same results. But here's my point. Throughout that time, autism rates continued to go up. So despite the fact that the vaccines themselves have gone down in terms of amount, autism rates have gone up. So there really is no link. Someone could make the argument that vaccines have actually been protective against autism. So it's really, there's nothing there. And I worry about that as a father, and I gave my kids vaccines. I think that's really important.
Night exclusive.
All right. Cool. So, this is, so what, did anyone take anything away from that video? Or what can we take away from that video? Anything?
I found it interesting that as the amount of vaccine reduced, the cases of autism increase. It's just funny to me, like the inverse relationship.
Right. And so, and that's perfect, right? Because to his point, he was trying to say that, the cause and correlation, for certain things, you may want them there, but they may not be there. Did anyone watch the vaccine TED Talk video that I put on Moodle? All right, cool. Megan, do you remember, I saw you shaking your head. Do you remember the graph that she showed up, kind of like a cause correlation, what she used as an example?
No, but it was 2 things that were like obviously not causational.
Right. So
it was like organic food.
Right, it's organic food. So she took a graph, right, and she showed organic food consumption in the United States has increased, over the last 30 years, and so has autism. All right. And so when you take those two pieces of data, you could technically argue that organic food is causing autism, which I think most of us would hopefully agree is not the case. And so his point was there can be danger in taking different data sets and just sticking them together. Now, one thing that I do want to touch on briefly and appreciate is that the news correspondent that was talking to Dr. Gupta is correct. There are adverse reactions to vaccines occasionally. And a lot of times it might be like 1 in a million. And, to his point, right, you're at more risk if you're taking, aspirin or Tylenol. You have a higher risk if you watch the TED Talk of, I thought it was funny, she showed a thing of getting injured at a dance party, or a higher risk, right, of getting in a car accident. But one thing I do, want us to always be mindful of when discussing vaccines versus anti-vaccination movement are, right, that 1 in a million, that is somebody, right? That could be somebody's kid. And so, I think when we approach this, approaching it respectfully and listening is really important. Because I don't agree with the second news correspondent that shouting the importance from vaccines at the rooftop and automatically labeling that group as wrong, even though they may be, is the right thing to do. I don't know about you, but whenever I got yelled at as a kid or told not to do something, I usually did the opposite or didn't listen. And so I think how we communicate and interact with people that are in the anti-vaccination camp is really important, right? And we can even relate this to the gospel. If you go up to somebody that is not a believer, you're just someone that's sinning, and you say, if you don't stop this, you're going to go to hell, how well does that conversion usually work? I would say not, right? And so how we communicate these things and how we interact with people that have different opinions with us is essential, not only in the vaccination discussion, but just in life. So I think there's a lot that can be learned from this debate. Ooh, this is one of my favorite things. Has anyone, and I guess you could even click ahead, but has anyone been paying attention to the COVID? I'll put Professor Perry on the spot. Professor Perry, what's their vaccine target kind of time from development to implementation for public use? Have you heard some of the numbers they've been spouting out?
When is the vaccine going to be ready?
Yes. What's their best case scenario time frame?
Well, I have heard there might be some doses ready in October, but mostly they're saying like next January or February, I believe.
Right. Perfect. Professor Perry's one of our local campus COVID-19 experts now. Thanks somewhat to my belief in her gifts, right? But she's right? And so the kind of, even the pipe dream here, the best case scenario, the Hail Mary, is more than likely 12 to 18 months, right, for a vaccine to go from development to use, right? And so vaccine development actually really started kind of gearing up in late February, early March, right? So about 12 to 18 months from them, best case scenario. I, like Professor Perry, have heard this fall. When we start to read through this list, I just don't believe that at all. That would be awesome. But one of the things you'll see, we'll get to here in just a second, is even if something's ready this fall, it's probably not going to be for mass consumption, right? And so let's walk through these kind of different points of vaccine development. All right. So first, you have to develop a prototype, or a fancy way of saying develop a target, right? What are you going after? Now, this is one of the things that we have been able to cut down the amount of time that it takes in this step significantly over the past couple of years. As computer modeling of proteins and viruses and other pathogens has improved, right, we're able to actually have a target. I was reading this, where I got this information from. A group had a target within a matter of hours, right? And so this used to take days, weeks, maybe even a year. We've really cut this down significantly. But even so, it's still kind of a predictive model. So it doesn't mean what you're predicting is correct, but it does give you something to go after, all right? I want to make a note here too. You see I have two things underlined: animal trials and phase 2 human trials. So on this slide, anything that's underlined is something that theoretically could be skipped in order to get a vaccine out to market more quickly, all right? Let's see. Glory, what would be your gut response to maybe, why would this not be good necessarily?
Well, if you skip steps, then like it could go like too fast, and then it might not be safe when they thought it might be safe.
Right. I mean, perfect. Right. It's a safety issue, right? So you're taking a risk. So, you know, to the timeline that I gave you, right, and Professor Perry mentioned too, I can almost guarantee you, if they're getting something out in 12 to 18 months, or if they're getting something out in October, that steps have been skipped, right? And that doesn't mean, if you're skipping these steps, that you're not concerned about safety. It just means that, right, sometimes there's downstream effects that we don't notice for a long time, right? And you are kind of eliminating potentially the possibility of catching some of those effects, right? So first, you have your target. Then typically what we've done in the past is we go to animal trials. And the animal trials, right, are basically a mock run in an animal model, right? We say, okay, first of all, is it toxic to the animal? And is it generating a protective immune response? All right? There's a good chance that if we inject a mouse or a rat with a small dose of vaccine, right, and it kills it, then we might think once or twice, right, about moving that to a human trial as is. All right, maybe you have to go back and rework some things, or potentially even start over, right? And so animals are really important for that initial safety test and that initial kind of proof of concept. Hey, this produces antibodies that are capable of neutralizing whatever pathogen we're interested in, right? You then go to phase 1 human trials, right? And this is not a number that you necessarily hold too hard. This is just kind of an estimate. Most phase 1 trials use anywhere from 20 to 80 people. All right, some may use 20, some may use 40, right? But somewhere in that range. And in this case, this is a really important safety test. You're injecting people to ensure that there's no major side effects. You're not, I mean, you can start to look at it, but the point of phase 2 is not necessarily, hey, are we making them immune? It's more of, hey, when I inject Glory with this vaccine, she doesn't have, seizures. She doesn't have, cerebral bleeding, right? She doesn't have some of these things occurring, right? So it's a big safety check. Phase 2, right, as we just mentioned, could be skipped. But this is when we typically move to a larger group, right? So maybe 100 plus, somewhere in there, to determine if the vaccine can protect, right? So we've said, okay, it's safe to inject people. Cool, that's good, right? They're not having adverse side effects. We then really are interested in, okay, is it actually protecting against a pathogen that we are trying to fight? Right. So then we go to phase 3. And here we really amp it up. Now we're going to hundreds or thousands of people to confirm that it can protect and to confirm that it's safe, right? All the while, people that were involved, right, in phase 1 and phase 2 trials can still be monitored to see if there's any adverse effects that are taking a while to appear, right? After your phase 3 trials are complete, you then go through a regulatory approval process. And this is where regulatory bodies, right, so like the FDA is a really important one that we have here in the United States. They review all of the clinical information that researchers have collected from these animal trials and these human trials, and they look at it and they say, okay, this is either safe for public use, so we can get started, or right, no, this is not going to work, we're going to shut it down, or hey, we're going to let you guys go forward, but we're going to ask that you keep on collecting data, right, from your different trials so we can feel assured that this is a safe vaccine. All right. You then have mass production, right? And this doesn't go as quickly as you think because there are strict quality control issues. We'll talk about this in the case in just a little bit. Some vaccines are made with something called attenuated pathogens. That means the pathogen is actually still alive. It's just weakened, right? And some of the ways that you do that are sometimes you heat them up, sometimes you add certain chemicals to them to weaken them. If you were to rush through this process, you actually would stand the chance of making a mistake and then injecting somebody with the regular pathogen, right? You don't want to do that. So this, mass production still takes time too, because the quality control regulations are really, really high. You then, right, will have public access to that drug, right? And where this becomes interesting is that often it's governing bodies that determine who gets first access, right? So to the vaccine that Professor Perry mentioned in October, right? I feel relatively confident that if they do have something, right, they're not going to have a dose for every person in the world or every dose for for people in the United States, right? They're just not. And so how do you determine who gets it? All right, so there's a lot that goes into this. I don't know, do I have it on the next slide? Let's see, or do I have it later? Okay, good, I don't have it here. So can anyone take a guess as to what the average timeline is? And I may have this, again, I think in a later slide. What's the average timeline for a vaccine to be developed? From that developmental phase, right, where you pick your target to when the public has access to it. And I'll mention this again later, but what do you think the average time is? Rachel D., what do you think?
2 to 3 years.
2 to 3 years. Okay. One other guess.
3 to 5.
3 to 5. Okay. So try 10. All right. 10 is very, very normal. Okay. And we might have one in 12 to 18 months, or maybe even October, which would be like November, December, January, February, March, April, May, June, right? I mean, I don't know, right? It will be incredible. I also do worry a little bit if we do have something ready that quick, right? How many of these steps have we skipped or kind of, right, maybe not done correctly, all right? So we'll revisit that a little bit. I have another slide here on COVID-19 in just a little bit. But let's talk about principles of immunization as it relates to vaccines. So I mentioned, right, you have attenuated vaccines, right, which are done with a viable microbe, right, that has reduced pathogenicity. And we'll kind of mention here in a minute some of the ways that you can do this, right? But this essentially is a pathogen that is capable of dividing, right, in a host, but, right, for a variety of reasons, it's not as pathogenic. But the body, right, the response to that attenuated pathogen is enough to generate a response that would be effective against the pathogen should it be seen by that host. Right? You also have inactivated vaccines. And in this case, you have a live microbe or live pathogen that has been killed, and we'll talk about variations to this. And then we inject, right, pieces of that pathogen into a patient, and that infers some type of immunologic memory and infers immunity. Okay? One of the other things that's cool about vaccines, right, is that they can be given in combination, right? So MMR, right, you're getting vaccinated for multiple things, right? If you've had your DTaP, right, they'll give that in combination. You'll get tetanus, diphtheria, and why am I drawing a blank? I'm drawing a blank here for a second on the other one. But right, we can combo these. Polio, which I believe Trent will present on. There are actually 3 polio viruses, and so the vaccine, when you get the polio vaccine, you get vaccinated for all 3 of those polio viruses. Streptococcus pneumonia. There are a lot of different variations of strep here, and so you get all of those variations in one vaccine, right? So we can group these things together. Right? So attenuated vaccines, right? Again, the basic idea of this is a weakened pathogen that should not cause disease, right? Emphasize should not cause disease. And these are great vaccines because this replication within the host induces, right, or generates a really robust immune response, right? The immune system has to actively engage with this weakened form of the pathogen, and that engagement leads to long-lasting immunologic memory, right? So these are really effective vaccines if you can make this work, right? And again, this basic concept of an immune response to a non-pathogenic strain will protect against the pathogenic strain, right? So the first kind of true vaccine for smallpox was actually using a cowpox virus, which was discovered by Edward Jenner. And cowpox just kind of gives you these little blisters that are full of pus on your hand. And that virus is similar enough, right? It's a pox virus, and it actually protects against smallpox. So sometimes it might actually be, right, a virus that's related to your target, but is not as pathogenic. So, you know, how do we get some of these weakened viruses? Well, in this case, we are interested in looking for mutations, or maybe even trying to induce our own mutations. Right? Mutations don't always make things more powerful. They could weaken certain pathogens. All right? Grow them in animal cells, right? So this is kind of a cool idea. So what you could do, right, is you could take the virus and, you know, I'll pick a random animal, right? Let's say for whatever reason you decide to choose a horse, right? Maybe you really like equines, and so you infect a horse with this pathogen. You then, you know, take serum from the horse, right? Or take a piece of tissue from the horse, right? Do a biopsy, and then you pull the virus back out, all right? And then you inject the virus that you've isolated or pulled out. I'll inject it into Amanda, right? I see her right here. Amanda, if I was using a horse as a study animal, what are you not?
I'm not a horse.
Yes, she is not a horse, right? Amanda's not a horse. And so it's this basic idea that the pathogen has gotten really good at, again, if we're using a horse as our model, causing infection or causing disease in a horse, all right? But Amanda's cells are different than horse cells. And so that pathogen is not nearly as efficient now at infecting Amanda's cells, but it's good enough that it induces some type of response so our immune system can generate that memory that it needs, right? Again, genetic manipulation, and so maybe you cause mutations, or maybe more than likely you actually remove certain components of the genome of that pathogen, right? We know, right, we've identified in a lot of pathogens what makes them pathogenic, and so you remove that set of genetic information while still allowing it to have the ability to infect its host, right? So those are just kind of some of the tricks that we do. We'll take a break here in just a minute. For attenuated vaccines, right? One of the advantages, and you can see how this might be a disadvantage a little bit later, But a big advantage is that they only take 1 to 2 doses, right? And so this is long-lasting immunologic memory. And so in some cases, you may never get vaccinated for these things again. And again, that varies person to person. So sometime, maybe when you're a little older, a little bit of an older adult, you know, a doctor might say, hey, we're going to draw some blood and look for antibody titers to certain for certain vaccines that you've had in the past. And if the antibody titers are low, they might say, hey, you know, get this vaccine again to make sure your body is ready in case you see it. One of the cool, again, and you could maybe see this as an advantage or disadvantage of attenuated vaccines, is because that pathogen is capable of infecting host cells and replicating, it's also possible, right, that that pathogen, that attenuated form, could be spread to other people, right? So, you know, if I gave one of these vaccines to Allie, right, and then she was hanging out with, looks like Lauren, Megan, Noelle, and Laura, right, it's possible that she could pass on that attenuated, weakened form of the pathogen to them, and actually, right, we get her to vaccinate them for us. All right, so kind of a cool little immunologic trick. Now, disadvantages. All right, because we are working with a pathogen that is alive, although weakened, right, immunosuppressed patients or immunocompromised patients could, right, actually respond to this vaccine like it was a normal pathogen, right? So their body might not be able to control it like a healthy individual could, right? And so you can't give a lot of these vaccines to patients that are immunosuppressed or immunocompromised, right? Should not be given to pregnant women, right? Pregnant women, if some of you choose, right, to have a family at some point, right, that's a lot of stress on their body. You also don't want you don't want the virus, even if it is weakened, interacting with the fetus, right? And so pregnant women can't receive these vaccines, right? As soon as they're without child, right, they could get them, but while they're pregnant, excuse me, they couldn't receive these vaccines, right? And these need to be refrigerated, right? And so this is a challenge for some of you that are interested perhaps in medical missions, right? Being some type of medical missionary. A lot of times the places that you may be going to may be very far away. And so keeping something cool over those long distances, when you travel from point A to point B, may be challenging. And maybe even when you get from point A to point B doesn't have power or doesn't have a reliable power source. And no reliable power means no reliable cooling or refrigeration. And so these can be challenging to deliver and preserve, especially if you're focusing on medical mission.
Why would they need to be refrigerated?
Why would they need to be refrigerated? Because, you know, and we can argue about viruses being, you know, alive or dead, but they technically are alive, right? And even some of these viruses or some of these vaccines, you need to keep them refrigerated to prevent any type of contamination of other microbes getting into them. Right. So, you know, just to give you an idea of some vaccines that are attenuated, right? So measles, mumps, rubella, rotavirus, which you guys hopefully got as small children, smallpox, which we don't really give anymore unless you're in the military, chickenpox, which you guys maybe had, yellow fevers, shingles you can get again when you're much later, right? So those are just a few common attenuated vaccines, right? So for inactivated vaccines, I'm sorry, this should be inactivated, disadvantage, and we'll talk about these and we'll take a short little break. These pathogens, right? It's good. They're unable to replicate. So we don't worry about the immunosuppressed or the immunocompromised quite as much with these vaccines, all right? The problem is when you're not able to replicate, the immune response may not be as robust, right? So your body may not be responding quite as strongly, right? So you have a limited immune response. And so I think I might have it in the next slide, I can't remember, but right, if you've had a vaccine and you've had to go in for a booster, right? The reason you're going in for that booster is more than likely that vaccine is inactivated, and you basically need to kind of generate an immune response that's strong enough for immunologic memory. And so sometimes that could include multiple injections. Right? The way that we help strengthen the immune response to an inactivated vaccine is something called an adjuvant. And so this enhances the immune response. So alum, so aluminum hydroxide and aluminum phosphate. This is one of the adjuvants. This is also something that, some folks in the anti-vaccination movement, would argue that you're putting something in the body that's not supposed to be there. If you go back and watch that TED Talk video, it's really interesting that there is, and this is just a really abundant kind of mineral molecule in our body and in the world. There's more aluminum hydroxide and aluminum phosphate in breast milk than in a vaccine. If any of you take Tums or antacids, there's a significantly larger amount of alum in that. And so, this idea that vaccines, right, are delivering a dose that's toxic just is not supported by the data and the concentration. All right. Oops. I was thinking there. So some inactivated vaccine categories, right? So you can inactivate the whole pathogen, right? So you just take, in this case, we have flu, polio, rabies. You inactivate the whole, these are all viruses, right? So one of the things you can do is you can basically treat them with formaldehyde, which is a chemical that basically causes cross-linking between proteins. And so you can then, right, inject that with the patient, and that induces an immune response. Toxoid, we actually talked about this in neutralization yesterday with antibodies. So diphtheria and tetanus, the vaccine is actually protecting against the toxins that are produced by these infections, not the actual bacteria. You then have subunit polysaccharide and conjugate inactivated vaccines. So here's some examples that you can categorize in here. So Hep B, if you've had that, HPV, which we'll talk about a little bit, whooping cough, bacterial meningitis. Most of you have probably had at least a majority of these vaccines. One of the ones that's really cool is a virus-like particle, and this is actually something that's being researched right now, even for the COVID-19 virus, right, SARS-CoV-2. And in this case, basically what you're doing is you're producing like a viral shell that has all of the proteins that are expressed on the surface of the capsid or the envelope of a virus, but it doesn't have any of the genetic information on the inside of it, all right? And so basically it's like an empty virus. And this is a cool potential future use for vaccines, but they're very complicated and very expensive to develop, right? And so parvovirus, right? There's a trial undergoing for that. Influenza, there's trials for influenza and viruses that I don't know if they're in use yet, but probably will be in use for Hep B and HPV are also underway. All right, but again, these are not living pathogens. They are not able to cause any type of infection. All right. So, again, advantage, I told you this, can't cause an infection. Also an advantage, a lot of them don't need to be refrigerated, right? Because they don't have live agents. Disadvantage, right? I said weak immune response requires boosters, so you have to go in multiple times. If you, for whatever reason, missed one of your boosters, then you often might have to start the whole process over again. And it is possible, right, in some patients for adjuvants to cause a strong inflammatory response, all right? And this can be one of the things that's associated with the negative side effects of vaccines. But again, that's relatively rare and varies widely from patient to patient. And so this is just from the textbook that I used to use, right? Talking about, hey, Emmeline, I'll come see you in just a minute. So these are some of the things that we had vaccines for, right? Some of them are more effective than others, but these are available to us. Some of the ones that I highlighted here, like diphtheria, whooping cough. I have it on the next slide. Yeah, you see measles, mumps, influenza, Hep B. These are things that we probably have all been vaccinated for. Interestingly enough, things like diphtheria, whooping cough, measles, we're seeing, and you can type this into Google, seeing higher numbers or the highest numbers of those illnesses that we've seen since the 1950s when some of these vaccines were introduced because, again, of the anti-vaccination movement. So you can kind of look through here and see, who they're recommended for. So as I mentioned here, if you look down here, shingles, right, adults age 60 and over, because it's typically not a problem in younger to middle-aged adults. Yellow fever, you're only going to get that if you're traveling to areas where it's endemic, right? So you might not have all of these. You probably have a good number of them, though. All right. So let's take a short break. It's 9:15. Let's come back 9:25-ish, and we'll pick up where we left off. All right. Oops. Right here, I see a chat here. Yes. Okay. So what the two options do here, right, is you basically are using like a measles virus that has already been attenuated. So it's a weakened measles virus, and you incorporate the gene for the S protein into the measles virus genetic information. And so you basically have little weak measles viruses that are going around in the host and causing an infection, and then generating the S protein in your body to respond to it. You have a non-replicating form, which basically is dropping off the viral protein at like a dendritic cell or a macrophage. The non-replicating form would require boosters, right, to achieve a full immune response, whereas that replicating version wouldn't. Protein-based is simply isolating the S protein, the spike, and injecting it into a person. If you remember from a couple presentations ago or whatever, you also have the M protein and the E protein, and there's even something called the N protein that are all surface proteins on the envelope of the COVID-19 virus, and those are kind of all targets also, right? And so just a lot of different possibilities. Oh, and sorry, also in the protein-based, that viral-like particle that we were talking about previously, that's a possibility basically, right? So you build the shell of the virus with the surface proteins, no nucleic information on the inside, and then you vaccinate that way. I know for at least the nucleic acid vaccine, this has actually never been used clinically. And so if this gets approved, it would be the first time that we've ever used this method to vaccinate on a wide scale. And with the electroporation or the protein delivery system, I'm not sure how viable it is, but it still is a really kind of cool, interesting way to go about doing this. All right. So I've already mentioned these, but what are some of the problems? Right. So we said the goal for this vaccine is 12 to 18 months, right, with the possibility of something being ready this fall, which I believe would be a miracle. I said vaccines typically take 10 years to be ready for public use, and if you want some interesting yet depressing information, so the previous record from developmental phase to availability for public use was an Ebola vaccine, and that was 5 years. All right. So I hope they prove me wrong, but you can understand why there might be a little bit of skepticism if they want it done in a year to year and a half, and the fastest that it's ever been done is 5 years. Even if you cut time off and skip steps, right, you could cut off 2 years from the process or 3 years from the process, and then you're still looking at a vaccine not being available for 2 to 3 years. All right. And by that point, it'll be interesting because there's still a lot we don't know about the virus. What will the level of herd immunity be from naturally occurring infections? We just don't know, right? We can't necessarily predict these things. It'll be interesting to see. And as I mentioned too, right, there's 90 vaccines currently, and this article was published in April, right? So it's not super at the end of April, almost early May. And so it's relatively up to date. And at that point, there were 90 vaccines being researched. I think there are probably even more than that now. But it's important that we're doing that many kind of possibilities, because fewer than 25% of all vaccines that start that developmental phase ever make it to the point where they can be used by the public, right? So, 75+% of those 90 vaccines that are being experimented with are going to fail to make it to public use. Now, that's also not guaranteeing that the ones that make it to public use are going to provide a super high level of protection, right? We just don't know. Maybe they protect 50% of the time, or maybe it's something where the immune response that's generated is only good for 6 months, and so you have to get it. We just don't know, right? So there's a lot of unknowns there. But when you certainly start to look at everything, can we do it in 12 to 18 months and have it be safe and have it be effective? I just don't know, right? That would be a marvel and a miracle of modern medical science. So we'll see. I'm not going to click on this link for you guys. We could watch it. It's the HPV vaccine commercial. I don't know if all of you have seen it, but it starts off with there's a version with a girl and there's a version with a boy. And it starts off with them like in middle school and, kind of saying, hey, if you would have known then, would you have got me the vaccine? And so this is caused by the human papillomavirus. Human papillomavirus. And in the United States, about a quarter of, our entire population, has had an HPV infection, So it's infected a lot of people. We have 14 million new infections each year, So this is a commonly passed around virus in the United States. And majority of cases, 90+% of cases are asymptomatic, and the infection is removed, or the virus is removed from the body within 2 years. All right. So most people, there's no symptoms, there's no adverse side effects. They clear it from their system and they're good to go. Right. Now, where we get concerned, is that in those 10%, some of the severe symptoms that we end up seeing later in life are cervix, vaginal, and vulva cancer in women. For men, there can be penile cancer. And for men and women, there can be anus or throat cancer associated with the virus. All right. And so, what are the CDC kind of guidelines or recommendations? All right. So if you get the vaccine, HPV vaccine when you're 11 to 12 years old, right? You get 2 shots that are 6 to 12 months apart, right? So some boosters there. If you're over 14, they recommend 3 shots over a 6-month period. And it's recommended for men before they're 26 and women before they're 21. Once you kind of hit this point, you could get the vaccine, but it's not common to get it. And this is from the CDC's website, right? You can get HPV by having vaginal, anal, oral sex with someone who's had the virus. It's most commonly spread during vaginal or anal sex. HPV can pass even when an infected person has no signs or symptoms, right? So kind of scary from the standpoint of you may be one of those 9 out of 10 where there's no symptoms, there's no adverse side effects, right? Your partner could potentially be the 1 out of 10, right? You just don't know. And in case you're wondering, right, so what's causing cancer? It turns out that some of the proteins that are expressed by the virus interfere with important proteins that help regulate your cell cycle. And so basically what the virus does is it stops the normal cell cycle, and there's no kind of checkpoints, and your cells, right, if you're at risk for cancer, divide uncontrollably, right, which is a hallmark of cancer, and that causes an issue in these patients, right? So the reason I bring up this vaccine is it can create, I guess, some interesting debates or questions about should we or when do we vaccinate. And this is a vaccine, right, that anti-vaxxers kind of use as an excuse to not get other vaccines, right? So again, and I don't, I think there could be a variety of opinions on this, but I'm fine, right? If someone kind of plays, devil's advocate. So why might there be, regardless of if you're an anti-vaxxer or if you're someone that gets vaccines, right? I could see people in both groups. Why would people potentially in both groups be opposed to this vaccine? No one wants to play devil's advocate?
Well, I mean, it seems like it's a vaccine that kind of is for people who are maybe like more sexually active.
Right. I mean,
Maybe
perfect.
so.
Right? Yeah, keep going.
No,
Yeah.
yeah.
And so, I mean, it's tough. And, again, if you're coming from a Christian perspective, even if that's not your perspective, right, there can be, right, that kind of assumption or that viewpoint that, hey, this vaccine is potentially promoting sex, right? And so do you do it? Don't you do it? And especially if you look at the normal vaccination period, right? 11 to 12-year-olds, all right? Or somewhere between 14 and 21 to 14, right? 14 to your early to mid 20s. I certainly can understand why as a parent, you're like, why would I get my 11 or 12-year-old? Why would I start a vaccine for something that's associated with sexual activity, right? And I'm not saying that's wrong or that's right, but right, this is a real question that has to be answered. So what's an answer or a rebuttal to that, right? How would you approach that if you were maybe a doctor having a conversation with a parent?
Well, I think I would say that you would rather be preemptive than once they get it, then you like take the vaccine or have to deal with the repercussions, because so just to be preemptive, just because they're going, I don't know if they will or not, but most people do have sexual activity later in life, so it's just preemptive.
Right, and I think that's from a medical standpoint, you're kind of as simple as, better safe than sorry, right? Yes, maybe this won't be an issue at all for your daughter or for your son, right? But maybe it will. And I think sometimes from a Christian perspective, right, some of those voices that are loud and screaming are like, well, you shouldn't have, this shouldn't be a problem because you're not going to have premarital sex, right? And I'm like, okay, maybe, right? I don't know, but I think we also have to remember as Christians, someone that has premarital sex is not automatically doomed right to the bowels of hell. And so even from a Christian perspective, you're like, if someone maybe makes a mistake in a situation, in a relationship, would you not want them to have protection from that virus and from potentially developing cancer later? All right. And so it's an interesting debate, right? This is not like other vaccines, right? This is not like polio, like a polio vaccine or a tetanus vaccine where you're just like, well, yeah, of course I'm going to get it, right? I don't, want my kid to get tetanus from stepping on a rusty nail. And so it's interesting vaccine, right? And it's not always so simple as, well, if a vaccine's available, you should just go ahead and get it, right? And so, unfortunately, from a Christian perspective, some people would argue, well, I'm not getting this vaccine because my kid's not going to, or they're never going to do this. And then unfortunately, the HPV vaccine sometimes gets lumped in with other vaccines. Well, if I'm not doing it for this vaccine, then I'm certainly not going to do it for these other vaccines, right? Even though that's not necessarily, I would say it's not really a good link between those two, right? But you can certainly see how for some situations or some vaccines, it's not as simple as just saying, hey, the vaccine's there, let's get it and let's do it. And so, I don't know. Just because there's some vaccines that you're like, should I get them or should I get them? Please don't think that makes you an anti-vaxxer. But there are decisions that have to be made. And I will say that, one of the things that's challenging for vaccines, and I'm a huge proponent of vaccines, that little small human that you saw sitting on my lap is up to date on all of her vaccines. In fact, she'll be getting her last vaccine on Wednesday. I think that's her last vaccine until she's like 3 or 4. So you kind of hit them early and hit them young. But sometimes in some cases it's not necessarily black and white depending on what the vaccine is for, right? And I certainly agree from a medical perspective. Do you want to give a vaccine for something that could possibly prevent cancer? And if you look at it from that perspective, I almost think the answer is always going to be yes. But then you can look at, how do you get the virus that causes this cancer? And then I think that could open up at least a conversation, right? So I don't know. Interesting stuff. I'm not saying that you're right or you're wrong for wanting to get this vaccine or not wanting to get this vaccine. I think, conversations don't hurt and are important to have for vaccines. Even the vaccines that you're like, why would you not get that, right? Of course, you should always get that. Having a conversation with someone about it may ease their mind, right, and some anxiety or tension that they have about getting it, right? So I think being open to these conversations is important for supporting the vaccination movement. Even if there are certain vaccines you're like, I don't know, right? So let's talk for a few more minutes on principles of immunoassays, right? So this is utilizing aspects of the immune system to our advantage in a research setting, all right? And so basically what we're doing is we're using antibodies to identify organisms, or using organisms to find antibodies. And so, sometimes what you see is you have an unknown organism here, and you have a solution containing antibodies, in this case to this Treponema pallidum. And so what you can do is you can take different antibodies and put them with this unknown organism to help you identify it, all right? Or you can have antibodies of an unknown specificity in a patient's serum, and then take a known organism, put them together, and see if they bind, right? And that allows you to identify either the organism or the antibodies that are present potentially in a patient. And this may reveal to you, or if you're going down to this bottom one, hey, I see Noelle right now. So I took Noelle's blood and I pulled the antibodies out of it, and you might have a couple different unknown organisms or known organisms, and you see if her antibodies bind to it, and whatever her antibodies bind to, that indicates, hey, Noelle might have an infection to that pathogen. All right. And so it's just kind of a cool basic principle, right? And then there's things that we can bind to these antibodies that give off a signal and allow us to detect the presence of a successful binding of those antibodies to whatever their target is. Well, that's a scary Easter bunny, huh? I know, right? So we actually, right, can develop antibodies to different pathogens and then use those, right, in the research setting. And so you have something called polyclonal antibodies. And again, this is that idea of having multiple antibodies that respond to different epitopes, which are part of different antigens, on the same bacteria, right? So this idea of you're injecting the whole antigen, you could think about injecting the whole bacteria, and different antibodies are going to respond to different parts of that bacteria, right? So you now have multiple antibodies, right, polyclonal, that are recognizing different aspects of the same pathogen, all right? You also, and the reason this happens too is you can have multiple B cells responding to, right, those different antibody, or to those multiple B cells responding to the same pathogen is going to produce different antibodies, all right? The problem with this approach is that these antibodies may bind to closely related organisms or antigens, right? And just a quick reference here, the reason that I have the scary Easter bunny on here is that oftentimes, or what we can do in a research setting is if we have an antigen and we want to generate a bunch of antibodies to it, we can actually inject, rabbits is a common test organism, so are mice. We inject them with the antigen that we're interested in developing antibodies to. They have an immune response. We then collect their serum and pull those antibodies out, and then we can use those antibodies in a research setting. All right, so we use animals to help us do this. All right, and so here's kind of the basic principle here, right? So you immunize a mouse, all right? You then pull cells from that mouse, right? So this is a terminal procedure, right? You isolate cells from that mouse, and hopefully some of these have responded to your antigen. You then take melanoma cells, and what's really cool here is you actually cause these cells to fuse together, and you produce a hybridoma, right? A hybrid is a combination. And so you basically make an immortalized B cell that produces antibodies that you're interested in studying, and then you can collect those antibodies. And this is how you get monoclonal antibodies, right? So you're taking out one B cell, right? Remember I said polyclonal. You could take the serum out of the animal. You could just take some of their blood, right? Not necessarily kill them. And then you just isolate all of the antibodies that responded to that one pathogen. But they're going to respond to different parts. Here, when you isolate this one cell, you're assuring that's a monoclonal antibody. This antibody is only going to bind to one epitope, right? That's part of one antigen. And so this increases specificity when you're using these in a research setting. This is just an example of what you might see I think, yeah, this is actually one of my old research slides here. So basically what I did here is I took poop from mice and made it into a liquid slurry. And then I ran that on something called a gel. And then I took serum from mice that had antibodies in it. And I probed this gel. So that means if the mouse had antibodies in its serum that responded to stuff in its poop, that's what's represented by these different bands here, okay? And so I'm basically using antibodies from the mouse's serum to see if it recognizes or reacts to something in its gut, right? And basically for my research, that indicated that something in the gut was able to leak across and activate the immune system, which is not what you want to happen. And you can do this a variety of ways. You can have enzymes that induce color changes that are attached to antibodies. You can have fluorescent dyes. You can have radioactive isotopes. So there's a lot of different ways that you can track these antibodies and their interactions with antigens. Let's see what I have left here. Yeah, okay. There's a few slides left. This is something called an ELISA. And there's two different ways that you can do ELISAs. One, you can essentially coat a, so actually this is even before this. So you have direct tests and you have indirect tests. And what you would do here is you could take either an unknown antigen or a known antigen. You are then marking them with antibodies that have some type of detectable marker on them. And if you have, in this case, some type of fluorescence, you have a positive result. No result, in this case, means that the antibodies that you put on did not recognize the antigen, and so you still don't know what it is. So then you would try maybe with a different set of antibodies that you know what the antibodies are to try to identify what this unknown antigen or protein is, okay? In this case, right, you know what the antigen is, and here you're taking a patient's serum. So this would be like, we go back to, I'll just keep on using Noelle, right? So she's infected with an antigen, right? Or we think she's infected with an antigen. We're not sure. So we know what the antigen is. We're not sure if Noelle's infected with it. So we take her antibodies and then we mix it with the antigen. And if they stick, right, we then take another antibody that has in this case, we'll say a fluorescent marker on it. And if they bound to the antibodies that are stuck, that indicates that yes, Noelle does have this pathogen in her system. If the first set of antibodies from her serum don't stick, then the secondary antibody can't stick, comes out to a negative result, and we're kind of back to the drawing board. Hey, Noelle does not have that pathogen in her, right? We use this similar concept for something called ELISA, right? Enzyme-linked immunosorbent assay. And in this case, you take a plate that has a bunch of wells in it. You can do one of two things. You can either coat it with the antibody to a known antigen, or you can coat it with an antigen that you know of. And then they give you a similar result, but it's almost kind of the opposite. So here you add what you think has the antigen in it. Here you add what you think has the antibodies in it, right? You then are tagging them with some type of marker. In this case, a lot of times these use little enzymes. These enzymes will cleave molecules that are in a solution and produce a color change, and then you can tell if you have a positive result or not. And these assays are really sensitive. You can also measure concentration. So you can determine the concentration of antibodies that you have present or the concentration of the specific antigen that you have present, if that's what you're adding. All right. So this would be like, you know, if, let's say I wanted to see, did Lauren have her MMR vaccine when she was little? And if she did, are there enough antibodies in her system that would provide protection? So what I could do is I could coat on the bottom of my plate the measles, a part of the measles virus protein or something like that, right? I could then take Lauren's blood, all right, and mix it. And if she has antibodies to the measles virus, right, they would stick to the virus that I put in the bottom of that well. And then I could detect them. Not only could I say, does she have it or not, I could determine how much of it she has present. All right, so kind of a cool little assay. This is something called FACS, fluorescent activated cell sorter. And basically what you do here is a really cool really cool experiment where you take a fluorescent marker and you attach it to cells, and then you run them through something called a capillary tube. They get shot by a laser, and if they fluoresce, that means your antibody is attached to the cell, and then you can detect it. And what's cool about FACS is some of them can then separate the cell based on what's attached to them. And so if you want to study a certain population of cells, you can mark them and tag them and separate them this way, right? And that's using antibodies to help you do that. And then, don't worry about this. If any of you have done ANP2, or those of you that will do ANP2, right, this is what we do for simple blood tests, right? Antibodies can cause something called agglutination. And so if you have the antibodies present, right, they will cause these cells to clump. So here the antibody for A-type blood, or the A antigens present, and so it causes agglutination, these cells clump. So that means this person is type A. All right. And then here, right, we're doing positive, negative. And so in this case, they're looking for the presence of group A strep. And so if group A strep is present, right, it will cause agglutination of these different antibodies, right? So basically, agglutination is a fancy word for clumping. And you remember how antibodies are helping you, right? Agglutination, they bring things together. They do that. They cause those clusters of bacteria to form so that you can remove them. And that's the same basic concept that you're doing here. There's also a little bit of indication, right? If you were to give somebody the wrong blood in a transfusion, this is what would start to happen in their body, right? And that's bad. Okay, and that is it. So I'm gonna let you guys, I'll move you right into your lab stuff. The lab assignment today is very brief. It's just on disinfectants. We normally would have a disinfectant lab. And so this is a brief disinfectant activity. While you guys are working on that, I'm gonna finalize your bacteria list. I'm gonna post that. And then you can even start working on that if you want. If not, if you rather leave after you finish your lab activity and get your study on, that's totally fine too, right? But that possibility will be open, and we'll talk even more about your unknown project in lab on Monday, but feel free to get a jump start on that as soon as I post this list. All right, cool.